ಕರ್ನಾಟಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿಯಿಂದ ನಮ್ಮ ಎಲ್ಲ ಶ್ರೋತೃಗಳಿಗೆ ಶುಭಾಶಯಗಳು. ಇವತ್ತಿನ ದಿನ ನಾವು 2022ರ ನೊಬೆಲ್ ಪ್ರಶಸ್ತಿ ವಿಜೇತರ ಬಗ್ಗೆ ತಜ್ಞರಿಂದ ಉಪನ್ಯಾಸ ಮಾಲಿಕೆಗೆ ಎಲ್ಲರಿಗೂ ಸ್ವಾಗತ. ನಾವು ಈ ವರ್ಷದ ಉಪನ್ಯಾಸ ಮಾಲೆಯನ್ನು ನಿನ್ನೆ ಶುರು ಮಾಡಿದ್ವಿ. ಇವತ್ತಿನ ದಿನ ನೊಬೆಲ್ ಪ್ರಶಸ್ತಿ ವಿಜೇತರು 2022 ಕೆಮಿಸ್ಟ್ರಿ ರಸಾಯನಶಾಸ್ತ್ರದಲ್ಲಿ ಅದ್ರ ಬಗ್ಗೆ ಮಾತನಾಡಲಿಕ್ಕೆ ನಮ್ಮ ತಜ್ಞ ಸ್ನೇಹಿತರಾದ ಅಮೆರಿಕದಲ್ಲಿ ನೆಲೆಸಿರುವ ನ್ಯೂಯಾರ್ಕ್ನಲ್ಲಿ ಹತ್ತಾ ಇರುವ ಡಾಕ್ಟರ್ ರವಿ ಕೋಡಿಹಳ್ಳಿ ಅವರು ಬಂದಿದ್ದಾರೆ. ನಿಮಗೆ ನಮ್ಮೆಲ್ಲರ ಕಡೆಯಿಂದ ಹೃತ್ಪೂರ್ವಕ ಸ್ವಾಗತ ಡಾಕ್ಟರ್ ರವಿ ಕೋಡಿಹಳ್ಳಿ ಅವರೇ. ಹಾಗೆ ನಮ್ಮೊಟ್ಟಿಗೆ ನಮ್ಮ KSTA ಸನ್ಮಾನ್ಯ ಸದಸ್ಯರಾದ ಪ್ರೊಫೆಸರ್ ಬಸವರಾಜ್ ಮೂಲಿಮನೆ ಅವರು, ಪ್ರೊಫೆಸರ್ AH ರಾಜಾಸಾಬ್ ಅವರು ನಮ್ಮ ಜೊತೆ ಇಲ್ಲಿ ಸೇರಿದ್ದಾರೆ. ನಿಮಗೂ ಕೂಡ ಸ್ವಾಗತ. ಹಾಗೆ ನನ್ನ ಸಹೋದ್ಯೋಗಿಗಳಾದ KSTA CEO ಡಾಕ್ಟರ್ AM ರಮೇಶ್, ಡಾಕ್ಟರ್ ಆನಂದ್ ಸೀನಿಯರ್ ಸೈಂಟಿಫಿಕ್ ಆಫೀಸರ್, ಶ್ರೀ ಉಮೇಶ್ ಘಟ್ಗೆ ಸೈಂಟಿಫಿಕ್ ಆಫೀಸರ್ ಎಲ್ಲ ಇದ್ದಾರೆ. ಸೊ ಈ ದಿನ ನಿಮ್ಮೆಲ್ಲರಿಗೂ ಸ್ವಾಗತ ಕೋರ್ತಾ ಮೊದಲಿಗೆ ಡಾಕ್ಟರ್ ರವಿ ಕೋಡಿಹಳ್ಳಿ ಅವರಿಗೆ ಸ್ವಾಗತ ಮತ್ತು ಕೃತಜ್ಞತೆ. ಇವತ್ತು ನೋಡಿ, ನಾವು ಈ ಭಾರತ ದೇಶದಲ್ಲಿ 11 ಗಂಟೆ ಇವತ್ತು ಬೆಳಗ್ಗೆ, ಆದರೆ ಡಾಕ್ಟರ್ ರವಿ ಕೋಡಿಹಳ್ಳಿ ಅವರು ಇರೋದು ನ್ಯೂಯಾರ್ಕ್ ಹತ್ರ, ನ್ಯೂಯಾರ್ಕ್ ಮತ್ತೆ ಬಾಸ್ಟನ್ ಹತ್ರ ಕನೆಕ್ಟಿಕಟ್ ಸ್ಟೇಟ್ ಅಲ್ಲಿ ಮಧ್ಯರಾತ್ರಿ ಕಳೆದು 12:15 ರಾತ್ರಿ ಆಗಿದೆ. ಆದರೆ ನಮ್ಮ ಕೋರಿಕೆಗೆ ಹೋಗೊಟ್ಟು ಬಹಳ ಆತ್ಮೀಯತೆಯಿಂದ, ಪ್ರೀತಿಯಿಂದ, ವಿಶ್ವಾಸದಿಂದ ಈ ಲೆಕ್ಚರ್ ಕೊಡಕ್ಕೆ ಬಂದಿದ್ದಾರೆ. ನಮಗೆ ಐ ಡೋಂಟ್ ಹ್ಯಾವ್ ವರ್ಡ್ಸ್ ಟು ಥ್ಯಾಂಕ್ ಯು ಡಾಕ್ಟರ್ ರವಿ ಕೋಡಿಹಳ್ಳಿ. ನಾವು ರವಿ ಕೋಡಿಹಳ್ಳಿ ಅವರು ನಮ್ಮವರೇ ಆದ ಇಲ್ಲಿ ಕರ್ನಾಟಕದವರೇ. ಅವರು ಕುವೆಂಪು ಯೂನಿವರ್ಸಿಟಿಯಿಂದ 2009ರಲ್ಲಿ PhD ಮಾಡಿದ್ರು ಇನ್ ಕೆಮಿಸ್ಟ್ರಿ. ಅಂಡ್ ದೆನ್ ಹಿ ವರ್ಕ್ಡ್ ಇನ್ ದ ಮ್ಯಾಸಚೂಸೆಟ್ಸ್ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಆಫ್ ಟೆಕ್ನಾಲಜಿ, ನಿಮಗೆಲ್ಲ ಗೊತ್ತಿರೋ ಹಾಗೆ, ಕೇಂಬ್ರಿಡ್ಜ್ ಫಾರ್ 5 years. Then he joined the pharmaceutical industry. Ravi has strong expertise in chemical biology and protein engineering technologies to facilitate biomarker discovery, chemoproteomics, synthetic biology, and drug development efforts. It's all tough areas in frontier sciences. His research has focused on translational regenerative medicine by applying click chemistry tools for developing quantitative omics. In fact, he is known as the omics man. Presently, he works with the Pfizer in USA, Drug Safety R&D USA, responsible for development and implementation of multi-parametric biomarker panels by adopting various cutting-edge technologies to understand the translation safety in pharmacology. He has worked, he also serves as a biomarker liaison and subject matter expert in rare disease areas. He's acknowledged, as I said, as the translational omics expert, and in the area of drug-induced liver injury, kidney injury, and so on. He represents the Pfizer and IQ Consortium, a non-clinical daily working group, International Consortium for Innovation and Quality in Pharmaceutical Development, and also participates in translation safety biomarker pipeline and CPAS predictive safety testing consortium effort. So like this, he's involved in various areas of frontier sciences to help the mankind. And we would have found no better person than Dr. Ravi Kodihalli to speak on the current year's chemistry Nobel laureates, Dr. Carolyn, Bertozzi, Dr. Morten, Meldal, Dr. K. Barry Sharples, for their work on development of click chemistry and bioorthogonal chemistry. And with these words, once again thanking Dr. Ravi Kodihalli, I would welcome him for the talk on this year's Nobel laureates in chemistry on behalf of the KSTA Karnataka. Dr. Ravi Kodihalli, please.
Sir, thank you, sir, for your kind introduction. So, can someone confirm? Can you hear me?
Yes, we can hear. Slides are fine.
Okay, thank you very much. Thank you to the organizers, and specifically Professor S. Ayappan and his staff, and Professor H.S. Savitri for the invitation to talk in the Nobel Prize lecture series. Also, I would like to acknowledge my institute, Massachusetts Institute of Technology, and my current employer, Pfizer, DSRD, Groton, United States, for allowing me to talk at this event. Today, I'm going to focus on this year's Nobel Prize in chemistry. and I'll take around 45 to 50 minutes to complete my talk, and I'll give a 10 minutes for Q&A session. On Wednesday, October 5th, 2022, most of us heard a sound click on every social media and a news channel worldwide. For the non-scientific community, it is just a sound while taking the picture or a photography. For a science student, it may be the word from their textbook. For a scientist, it's a joy. You read it and you did it in the research lab. On that day, the Nobel Prize Committee announced the name of 3 scientists, Professor Carolyn Bertozzi and Professor Sharples from the United States and Professor Morten Meldal from the Denmark are the winners of 2022 Nobel Prize in Chemistry for the development of click chemistry and bioorthogonal chemistry. Click and bioorthogonal chemistry. On this title, you may see 2 separate words. One is click, another one is an bioorthogonal. The click chemistry revolutionized the chemist option for creating the desired molecules. At the same time, the bioorthogonal chemistry made it possible to monitor the chemical processes inside the living cells without harming them. All these discoveries in this area transfer the synthesis of everyday household materials and life-saving pharmaceuticals or drugs, and it created a new opportunity or a new venue to treat the patients in clinic. Then the question is, what is click chemistry? What is the significance of click chemistry? When you think about the natural products, these are the molecules or compounds that those are from the plant and animal origin, which may use every day directly or indirectly. They have been tested for their medicinal properties, and some of them are showed a tremendous potential, for example, antibacterial or antifungal activity. The beauty of these molecules is their complexity and the diversity. Even chemists are often shocked by them, and they mimic nature or natural molecules by artificially synthesizing them in the laboratory settings. Still, there is a great difficulty in making these complex molecules. In nature, biomolecules are created just like this, starting with a reagent A. Then a sequential reaction from 2 to 4, forming a many intermediates. And finally, that end up with a product and then byproducts. So when chemists try to make these same products in the lab or in a synthetic system, we need to having in many unwanted materials and byproducts. So no use, and some are harmful to the environment and more expensive to make. But in nature, they are all eaten up by the plants or from the plants. They used for the different purposes. The 3 Nobel Prize winners of 2022 put forward a great alternative called a click chemistry. And here is the way those works. Assume this is a reagent 1 and this is a reagent 2. We click them or join them together, forming a product. The advantage of this reaction is that there is no intermediate, no byproducts, and these are straightforward reactions, which is why these reactions called click reactions, an easy way to make the chemical molecules. The great thing about these reactions is that those are efficient, they occur at mild conditions, and to bring the 2 different molecules together by clicking them one to other as one does with a seat belt. Let's discuss one by one about the three scientists and their history, why they got the Nobel Prize. First is Carol Barry Sharples. He received his PhD from the Stanford University in 1968. After his postdoctoral work, he joined the Massachusetts Institute of Technology in 1970, and he worked for 17 years. Then in 1990, he moved to the Department of Chemistry at Scripps Research Institute, United States. And he is a two-time Nobel laureate. For his work on the chirally catalyzed oxidation reactions, he received a Nobel Prize in 2001. He is the only living person awarded two Nobel Prizes, as only 5% in history received them. In 2001, Sharples wrote a review article in the Journal of Chemistry. In that article, he coined the term click chemistry, arguing that chemistry needs to move towards more straightforward reactions with less waste in the form of an unwanted byproducts. According to him and the paper, just a handful of good reactions are needed to assemble the extensive number of highly diverse organic molecules. Then Professor Morten Peter Meldal. He received a PhD in chemical engineering from the Technical University of Denmark. Then he was a postdoctoral fellow in the organic department in the same university for some time. Then he moved to the MRC laboratory at Cambridge University, then at the University of Copenhagen. He did three postdoctoral work. Then currently he is a professor at University of Copenhagen, Denmark. According to Nobel Prize website, he developed a smart and efficient chemical reaction, the copper catalyzed azide alkyne cycloaddition. This is now a widespread use and is utilized in developing pharmaceuticals, for mapping DNA and creating the new materials. Finally, Carolyn Bertozzi. She completed her undergraduate from the Harvard University and then PhD in chemistry at the University of California, Berkeley in 1993. She is currently working at Stanford University. Interestingly, Bertozzi has taken this click chemistry to a new dimension and invented a field of bioorthogonal chemistry, which allows the researchers to chemically modify the molecules within living systems. And Professor Bertozzi coined the term bioorthogonal in 2003 to describe the reactions that do not interact or interfere with the cells in a biological system. The beautiful illustration of three Nobel laureates about the click chemistry by Johan Jamsted from the Royal Swedish Academy of Sciences is shown here. On the left side is the Barry Sharples, where he pioneered the concept of click. On the right side, the Professor Meldal, who performed or applied the copper catalyzed click chemistry. In the center, Professor Bertozzi coined the term bioorthogonal, like a ring structure. I will talk about those things in the next few slides. Here is the timeline of the entire award-winning discovery. In 1990s, Professor Sharples thought about a streamlined way of chemical synthesis. In 2001, Sharples summarized the concept of click chemistry in an Angiochemy paper. And the same year, Professor Meldal discovered the azide alkyne cycloaddition reaction, and he done independent work. But in 2003, Professor Bertozzi introduced an idea of a bioorthogonal chemistry and to monitor the cellular process. And in 2012, the entire effort won the Nobel Prize. It took almost 3 decades to win the prize. Next few slides, I will provide you more details about these three scientists' works. First is the Professor Sharples. The compounds, those are derived from the natural sources such as plants, animals, and microorganisms, have a greater and enormous impact on medicine and health, and we often use them to treat the disease and liver injuries, and still it is exist in my villages in Karnataka. Last few decades, these natural products have taken a role in the drug discovery and drug development. The modern tools of chemistry and biology, in particularly various omics technologies like new chromatographic techniques or HPLC methodologies, now allow the scientist to extract individual components and test them against the many devastating diseases. And they showed a great promise. But the synthesis of natural products in the lab constantly facing the many challenges because of their complex nature and the lengthy synthetic routes, and also the time-consuming workup protocols and large environmental footprints. Here is an example of meropenem, an antibiotic. It took nearly 6 years for the large-scale industrial production after many chemical modifications. Another example is Crixicol, an AIDS protease inhibitor. It took a multiple years to synthesize in a large scale. Professor Sharples is the first person to coin the term click chemistry in 2001. In biomolecules, there are bonds between the carbon atoms. But in synthetic system, these carbon atoms lack the motivation to form a bonds with each other. So he thought that we need to activate them artificially, and he suggested the functionalization of these atoms with nitrogen and oxygen, or creating a carbon heteroatom linkages so that they can click together in a predictable way to make easily controllable. According to him, for a chemical reaction to be called click chemistry, it should occur in presence of oxygen and water because those are cheap, inexpensive, and environmental friendly. It's a concept in which molecules are simply, quickly, reliably, and repeatedly joined together in which the same way the way as a seatbelt clip into its buckle. According to Professor Sharples, click is the pure fusion process, and which means that the combined formula of the reactant is equal to the formula of the product. In that way, click reactions are very broad in chemistry. Many reactions fall into that category. Example, cycloaddition, nucleophilic substitution, carbonyl condensation reactions fall into that category. Next is the contribution from the Professor Morten Meldal. Actually, in 2000 and 2001, he was developing a methods to combine the azide, a molecule that has the 3 nitrogen atoms in a row. On the other side, in an alkyne, a molecule which is in 2 carbon atoms and are bonded together with a triple bond. By themselves, these 2 building blocks are not very reactive. If you mix them together, they are very slow to react and yield a mixture of products. But the unexpected reaction twist happened when they added a bit of copper to the mix. The reaction accelerated dramatically and led to a stable product known as triazole. This copper catalyzed reaction immediately gained enormous interest across the chemistry and related field because triazoles, which is a versatile chemical that has many applications in pharmaceutical, as an antifungal agent, antibacterial agent, in making the dyes, in agriculture as an fungicides, then building blocks of many chemicals. According to Nobel Prize Committee, we can say it is still the crown jewel of click chemistry, that is called copper catalyzed azide alkyne cycloaddition. Finally, Professor Carolyn Bertozzi's contribution. Professor Bertozzi took the click chemistry to a new dimension and began using it to map the cells. Our bioorthogonal concept or reactions are now contributing to more targeted cancer treatments among many other applications, and most recently she applied to the even COVID-19 patients as well. Then question is, what is bioorthogonal reactions? A bioorthogonal reaction is a reaction taking place within the living system which does not interact or interfere with the native biochemistry of the system. And these reactions which occur without disturbing the normal chemistry of the cell have a huge implication for a biochemist or in the field of biochemistry. Assume this is a cell. In the center, there is a nucleus and around it cytoplasm. As we know, glycans are the complex carbohydrates built from the various sugar present on the cell surface. They play an important role in many biological processes such as immune regulation and intracellular interactions. To understand this glycan biology, how cell work, and to map the genes and proteins, Professor Bertozzi fed the cells with a modified sugar with the azide group on it, so that this azide could serve as a molecular handle. Once modified sugar was incorporated into the glycans on the surface of the cells, she used a ring-strand or ring-shaped alkyne molecule to perform a click chemistry. It is an independent of copper, which can be effectively used inside the biological system without any toxic effects. Interestingly, she attached this ring-shaped alkyne to a fluorescent molecule where it can emit the light. Using this ring-promoted click reaction followed by the emitted light, she revealed where the glycans were hidden in the cells, and this click methodology allowed her to track the glycans on the cell surface. Here is an example from our 2007 Proceedings of National Academy of Sciences USA paper, where she showed it. She tracked the glycans on the cell surface by forming a click chemistry. And that is indicated in the green in this picture. The cell nucleus is colored in the blue. Currently, some of the clickable antibodies now in the clinical trials. And Professor Bertozzi's contribution helped the scientists across the globe to map the genes and the proteins to track the biological processes effectively. Now, I'm going to talk about the applications and the future aspect of the click chemistry. Click chemistry has a dramatic and diverse impact on many areas of modern chemistry. As I discussed in my previous slide, tracking the movements of molecules through and across the cells is just one of many applications of click chemistry and bioorthogonal chemistry. A major advantage of this technique is that they don't introduce unwanted byproducts into a reaction mixture. They function with a clean efficiency that allows the scientist to carefully craft the complex molecule for various purposes. If you look at the literature, there are a growing number of applications are found in various fields. And even in the case of drug discovery to the material sciences. Here I listed a few, and these are the sections in terms of a biomedical applications. And the combination of click chemistry with a high throughput screening or molecular assembly systems provide a rapid, efficient, and reliable tool for the discovery of bioactive molecules. And this click chemistry become one of the most powerful tools in chemical biology, drug development, DNA sequencing, and synthesis of smart materials, and many technology driven innovations. I'm going to talk on those things in next few slides. And for example, in the proteomic application. And here is next few slides, I'm going to talk about my own work and where I use this chemistry to develop a technology during my MIT days. The first technology I developed using this click chemistry is called an activity-based protein profiling technology using a mass spectrometry. And this technology integrates the strength of both chemical and biochemical disciplines. During the drug discovery process and disease diagnosis, it's always important to understand the critical information and real-time monitoring of catalytic activity of the enzymes, especially in a disease model or in case of a disease context, so that those enzymes can be used as therapeutic targets. Also, they can act or serve as disease monitoring biomarkers. And let's go through into the activity-based protein profiling technology first. For this application, and for the technology development, a small molecule probes are very important. Those can target the enzymes of interest in a biological matrix. Because in a matrix, in a biological matrix, means it may be serum or plasma or a tissue lysate. In those matrices, you can find a thousand number of proteases. For that reason, first we need to design a chemical or a small molecule probe that consists of two units. First unit is a reactive group, or an warhead we call them, which can target the active site of the enzyme of our interest. The other end of the probe is linked to the clickable group. In this case, it is attached to the alkyne group to facilitate a click reaction. In principle, most of the time, these warheads or probes are the inhibitor of the enzyme. Another unit is the reporter group. Those are most of the time, those are the fluorescent molecules. And those are attached here in case of for the azide group to perform a click reaction or perform and so that when we combine together, they can facilitate the target fishing. In this case, we have fluorophore. We can easily visualize once performing a click reaction on the STS page gel, and it's one of the rapid screening methodology and commonly used laboratory technique. In the other way around, if the reporter group is in biotin and it is attached to the azide, after performing a click reaction, we can pull down on the streptavidin beads, followed by the trypsinization. We can simultaneously identify and quantify the enzymes. Those are targeted by our probes using a mass spectrometry. By combining this click chemistry and a mass spectrometry, we can easily identify the undruggable proteins or enzymes. The interesting thing is that this chemical biology technique can provide an unbiased global and quantitative analysis of protein binding partners, and it is currently used to determine the functional state of enzyme directly in the native forms. Also, this technology reveals the complex physiological and pathological enzyme substrate interactions at molecular as well as cellular levels. It is the class of enzymes or proteins we are interested in developing the technology by using a click chemistry and a mass spectrometry. Those are called matrix metalloproteases, also called MMPs. Those are secreted or membrane-bound endopeptidases and have been shown widely known for their ability to cleave the wide range of extracellular matrix components and thereby contributing to the tissue turnover. And interestingly, this expression of these MMPs are strictly controlled under normal events, but on the other hand, their abnormal activities or overexpression or overproduction is harmful most of the time and often noted in many disorders such as neurodegenerative, cardiovascular, and arthritis and digestive diseases, and some in some cancers as well. And we are interested in profiling the active MMPs in an osteoarthritis and rheumatoid arthritis patients. As we know, osteoarthritis is a degenerative joint disease found in elderly people that is triggered by the accumulated mechanical stress to the joints, leading to the destruction of cartilage, whereas rheumatoid arthritis is a systemic inflammatory diseases of joints, and still today it's not fully understood. Here is the structural illustration of a joint. In disease condition, in case of an osteoarthritis, the pro-inflammatory cytokines and such as IL-1 beta, IL-6, IL-17, TNF alpha are most of them they will be elevated, and those elevated cytokines are known to activate MMPs and their family members that lead to the many anabolic as well as the catabolic activities that initiate the cartilage loss, bone remodeling, and inflammation of synovial membrane. The identification of the specific MMPs in that environment or in that matrix is very important to treat the disease, because there are 30 different matrix metalloproteases are known based on the literature. Still today, we don't have a specific method or a technology is available to monitor this catalytic activity of metalloproteases in a disease condition. First, to target these MMPs, we designed a chemical probe with a warhead that targets the active site of the matrix metalloproteases. Other end of the probe was linked to the azide group, that's called N3. Then we added these probes into a biological matrix of proteins. Then the probe can bind to only the active MMPs, since it's designed to target a specifically for the matrix metalloproteases. Then we use a strained alkyne with a solid support to perform a click reaction or a click chemistry and to pull out the probe-bound metalloproteases from the complex mixture. When we perform the mass spec analysis, we selectively identify some of the MMPs and its members as in therapeutic targets or in disease biomarkers. Finally, the combination of click chemistry and mass spec, we are able to develop a technology that can selectively identify the proteins of our interest in a disease model. And now it can be applicable to the many disease areas where MMPs are activated. After achieving this goal and gaining the confidence in the click chemistry based activity-based in protein profiling of MMPs in a disease model, we look behind this technology and took this activity-based protein profiling method into the next level and we developed it and we applied it in a very user-friendly technology that's called an MAMBI technology, or we call them Metalloproteases activity multiplexed bead-based immunoassay technology. It is very user-friendly. It doesn't need a mass spec because, as we know, mass spec needs highly experienced personnel and it's also expensive. We cannot use in the hospital settings. It's because of its expensive nature. But this MAMBI technology can be usable in any research labs or hospital setting to monitor the activity of proteases. Here is the way it works. As described in my previous slide, MMPs are zinc-dependent endopeptidases responsible for the regulation of normal and diseased cellular functions. Most MMPs are produced in the zymogen, as a latent form. Some of them are activated internally by internal process because they themselves chew each other. And some of them are inhibited by the endogenous inhibitors because in a biological system, there are a couple of MMP inhibitors still exist. Those are called tissue inhibitor of metalloproteases. Here we use the activity-based probe that is combined already to the biotin. We first incubated this activity-based probe in a biological matrix. After the samples were labeled using an activity-based probes, since these probes are directly targeted to the active metalloproteases, then they will go and bind to the active site of these metalloproteases. Then we pull down all the MMPs, including zymogens, inhibitor bonds, onto beads that are coated with the MMP antibodies. Those antibodies can capture all the metalloproteases, including the probe-bind ones and the inhibitor-bound ones as well. After extensive washing, clean up the samples. Since our active proteases are bound by the chemical probe, those are linked to the biotin, we visualize those biotin using a step-tagged in PE. In that way, we quantify the active metalloproteases because the step-tagged in PE is not going to bind to any of the other or inhibitor bound or a zymogen bound metalloproteases. This is the table shows that the assay is very sensitive. It is one of the best methodologies to identify the active MMPs, and this assay requires very significantly very less volume. Also, we can, as you can see, most of the MMPs are detected at picogram to the femtogram level. Also, we can detect and apply this methodology into the many biological fluids and cell cultures as well as tissue lysate. And now this technology is transferred to the commercial vendor, and soon it will be available for the researchers. The reason I showed this data is because we started by developing one technology by using click chemistry and a mass spectrometry, which led to another user-friendly method that's called MAMBI method. Now we are testing this technology in organ on a chip models or human body on a chip system to see the potential of this application in the field of personalized medicine. This is a classic example of how click chemistry discovery are an important accomplishment made by Professor Sharples, Professor Meldal, and Professor Bertozzi impacted the many researchers around the globe to think behind the wall to achieve and develop technologies to improve the patient's health. Finally, I would like to summarize the entire story in 3 statements made by this great scientist. First is Professor Sharples. He made a statement in 2019 that inspires every researcher. As it reads, asking good questions is vital. Strong interference, multitude of working hypotheses, and the periodic table will never let you down. That's what he did it in his entire life. Functionalizing the molecule using an heteroatoms such that those are readily available for click reactions. According to him, the reactions need to be perfect. In other words, they need to produce 99.99% heat. Using a click chemistry, we can achieve that goal. Even I perform those reactions in the laboratory setting in a biological system. It's possible to get the around 99.99% heat. And the second is the Professor Meldal. According to him, it is an extremely difficult to do one kind of a chemistry in the presence of another kind. What he has done something which is completely orthogonal to all the existing chemistries and something we cannot imagine. An azide, a molecule with the 3 nitrogen atoms bound to a bound in a straight line, and an alkyne containing a triple bond between the carbons. And by using a copper, he made a ring, is a triazole, copper catalyzed azide alkyne cycloaddition reaction. That chemistry now can be used for making the bridges in peptides, protein structures to stabilize their properties. And it indicates we don't need many reactions. Just a handful of good reactions are enough to assemble the vast number of highly diverse organic molecules. Finally, Professor Bertozzi. Soon after the public announcement on October 5th, Adam Smith from a Nobel Prize committee called Professor Bertozzi. It's in the early morning in the United States. She was in her house in California, and she was talking to the Adam Smith on the phone, and Adam Smith asked her how you are feeling. That time she mentioned one thing, that I cannot help but think about all the women that came before me that didn't have an opportunity to be recognized, because she's the 8th female Nobel Prize laureate in chemistry. And according to her, chemistry is an exciting area of science for people who want to have an impact, because she'd done it. And she's more optimistic about how science and the culture of science is trending. Finally, I'm one of the fortunate guy to work in this area of this great scientist discovered in the same department as Professor Sharples, because he was a professor at Department of Chemistry at MIT. And another takeaway from this year Nobel Prize is that 2 things. One is a woman won the Nobel Prize. That's the first thing. And the second thing is that the green chemistry, where we are talking about a lot on the climate change, all those things. Is the green chemistry is having a huge impact, because it cut down the many multiple reactions in a chemical synthesis. Just I talked about in just biomedical applications. That's my area of interest. If you talk to the chemist, or if you look into the literature, you will get in many examples and many applications. Finally, I would like to acknowledge my mentor, Professor Steve Tenenbaum. Is the professor of biological engineering, chemistry, and toxicology, and who gave me the opportunity to work in this area. Professor Linda Griffith. She's the professor of biological engineering and mechanical engineering. And Professor Alan Gorzoski is a professor of electrical, mechanical, and biological engineering at MIT. And all my colleagues who worked with me in developing these technologies. Thank you so much for your attention. I'm happy to take any questions if you have.
Thank you very much, Dr. Ravi, for this scintillating talk. Amazing, in these 45 minutes to 50 minutes, you have summarized, several things. You mentioned about the Nobel laureates, definitely for the field of chemistry in 2022. And in the course of the deliberations, you brought out the significance of all that's happening in the area of green chemistry. And only you could have done this job so elegantly. And I do not have words adequate at this stage to thank you. We also have with us the Vice Chancellor of one of the universities in Bangalore, Dr. K.N.B. Murthy, as I can see. So several of them have thoroughly enjoyed your talk. We'll have some more discussions. As you said, you are ready to take a few comments and questions. So at this time, it's time for our esteemed audience to have some remarks, opinions, any questions that you have. Yeah, open for discussions, please. Any remarks from our esteemed colleagues?
Professor Ravi, hello.
Yes.
It was a beautiful talk. I'm from physics area, but I really enjoyed, you beautifully brought out the importance of this click chemistry. And I think it's going to open a new path for finding many applications which you listed, particularly with regard to DNA also you mentioned. How important that is, with regard to AIDS or cancer. You also mentioned about COVID, any other things. It's the most easiest path or that can be pursued in our country, this technology.
So, yes, there are tremendous technological advancements are taking place. I will come back to your questions later.
Yes.
The thing we are testing now in microphysiological systems, that's what I brought up that point, like for example, human body on a chip or organ on a chip models. That's a personalized medicine, because in a cell culture, we do at the common laboratory settings, this 3D environment, what we are doing, that is taking our own cells and putting onto the microphysiological system. And there we are testing. If that hypothesis is successful, I'm pretty sure that can easily transferable to the clinic. Then we can take any patient cells, if it is a cancerous cells or any other cell types, and we can take into those 3D systems and we can test them. And we can identify the disease monitoring biomarkers or a target of our interest. We can directly treat those patients within particular class of drugs. There is a possibility. And the second thing is that taking these technologies to the our country, I'm pretty sure it's possible, because a lot of scientists, even including me and everybody, even a lot of people in the Indians of Science, ISARs, a lot of scientists are doing this kind of work already. They started with a click chemistry now, and now they are going for the DNA modification, and also the Professor Bertozzi's work using an bioorthogonal chemistry. They are implementing all those things, but it's taking some time, because the technology, it's not very easy, because as I mentioned, mass spec technology, when we started, it's a so complex process.
Yeah.
When you move to the second technology, I mentioned MAMBI technology, it's a bit easier. First time is always a challenging, but it will be there in a few years. I'm confident about that, because there are a lot of highly talented scientists around India. They're working on the similar kind of a technology. I talked about only the biomedical side. If you think about the chemistry or a polymer science, and we have the JNCS or their expert. They have a lot of experience in the polymer synthesis, and they're applying these kind of tools they're implementing in their synthetic protocols.
Okay. Thank you very much, Professor. So nice. Very great. We are grateful.
Thank you.
Thank you, sir. Any other comments, remarks from our colleagues? Any questions? Yeah. Okay. So thank you very much. Now we will have our words of thanks formally also, but I cannot contain myself, in acknowledging your wonderful talk, appreciating, your acceptance of our request at this odd hour. I must repeat, it may be around 1:30 or so, or maybe nearing 2 o'clock after midnight in the US. But then you look so cheerful and fresh as in the morning, Dr. Ravi Kodihalli, and person working with an elegant company, global company of Pfizer, coming to our academy for delivering this talk on the current year's Nobel Prize. We are really fortunate, and no words to thank you enough, Dr. Ravi Kodihalli. ನೀವು ನಮ್ಮ ಜೊತೆ ಬಂದು ಮಾತಾಡಿದ್ರಿ, ನಮ್ಮೆಲ್ಲರಿಗೆ ಶ್ರೋತೃಗಳಿಗೆ ನಿಮ್ಮ ಭಾವನೆಗಳನ್ನ ಹೇಳ್ಕೊಂಡ್ರಿ. ಇನ್ನೂ ಒಂದೆರಡು ಏನಾದ್ರು ಸಂದೇಶಗಳಿದ್ರೆ ಕನ್ನಡದಲ್ಲಿ ಕೂಡ ಹೇಳಲಿಕ್ಕೆ ಇಷ್ಟಪಟ್ರೆ ದಯವಿಟ್ಟು ಹೇಳಿ. ಇಟ್ ವಿಲ್ ಬಿ ಸ್ಫೂರ್ತಿ ಬರುತ್ತೆ ಎಲ್ಲರಿಗೂ. ಡಾಕ್ಟರ್ ರವಿ ಅವರೇ.
ಖಂಡಿತ ಸರ್, ತುಂಬಾ ಧನ್ಯವಾದ ಸರ್. ಈ ಒಂದು ಇದರಲ್ಲಿ ಮಾತಾಡ್ಲಿಕ್ಕೆ ಅವಕಾಶ ಕೊಟ್ಟಿದ್ದಕ್ಕೆ ತುಂಬಾ ಧನ್ಯವಾದ ನಿಮಗೆ. ಅಂಡ್ ನಾನು ಇನ್ ದ ಬಿಗಿನಿಂಗ್ ಅಲ್ಲಿ ಹೇಳಿದ ರೀತಿ Professor Savitri madam, actually one of the great scientists, as a woman, as a scientist, she's amazing. And also it's not easy in this environment to achieve that level of standard in science, and she done it. And those are the real example for us, actually. So when we think and when we are interact with them, and that inspires a lot. As I told in the beginning, if you want to achieve the great thing, what we need to do is that just listen to the parents and the teachers. Those are the two things I followed. I'm pretty sure all of you at this level, you followed those things. I think in the next generation, if they followed those traditions, I'm confident all of us will achieve the greater heights. And in this way, I would like to acknowledge Professor H.S. Savitri, Professor M.R.N. Murthy, Professor L. Shashidhara, and Dr. M.R. Sitalakshmi, and all this. I interacted those things. Those are my well-wishers, and they taught me great things as a teachers, as a mentor. So I don't have any words to say, and thank you to you and all the colleagues here, and and everybody, actually. So for this invitation, and I'm happy to be part of this event, and I hope I convinced all of you on what is click chemistry, what's the applications, how the world is thinking, why when they are deciding the Nobel Prize, why they are giving the prize. Because as I mentioned, it's a 30 years effort, and how we are using in the current technology development. Just one example I gave, and there are plenty to come. I'm pretty sure we can make use of all this technology for our better health and for the humankind. Thank you very much.
ಡಾಕ್ಟರ್ ರವಿ, ತುಂಬಾ ಧನ್ಯವಾದಗಳು ನಿಮ್ಗೆ.
ಧನ್ಯವಾದಗಳು.
ನೀವು ನಮ್ಮ ಎಲ್ಲ ಶ್ರೋತೃಗಳಿಗೆ ಮನಮುಟ್ಟುವಂಗೆ ಇದನ್ನ ವಿವರಿಸಿದಿರಿ. ಮತ್ತೊಮ್ಮೆ ಬಹಳ ಬಹಳ ಧನ್ಯವಾದಗಳು. ಓವರ್ ಟು ಡಾಕ್ಟರ್ ಎಂ ರಮೇಶ್, CEO of the Academy, for his words and words of thanks. Dr. Ramesh, please.
Namaskara. Good afternoon. Good evening. Morning. Thank you very much, Dr. Ravi Kodihalli, for this very enlightening and very lucid presentation on the works of Nobel laureates, Professor Carolyn Bertozzi, Professor Morten Meldal, and Professor Barry Sharples. Indeed, many of us were wondering what is click chemistry, what is orthogonal biochemistry, but you explained it so lucidly that we could pick up some points from your talk. I'm not a chemist, but I have studied chemistry in my bachelor's and master's long ago. I've forgotten, but your talk was very, very lucid and easily understandable. I'm sure many of those who have attended the talk would also join with me in appreciating your talk. On behalf of KSTA, all the audience present here, I'm very immensely thankful to you for your presence, even though it's a late, early morning, very early morning in the US. You took time from your busy schedule and delivered this wonderful talk. On behalf of KSTA, we are very grateful to you. And when you come to India, we'll be very happy to organize an offline talk by you on advances in chemistry in the future. Thank you very much. I'm also
Thank you.
Chairman Professor Ayappan sir for guiding the program. I'm very thankful to our esteemed members of KSTA, Professor Mulimani sir, Professor Raja Saab sir for joining this talk and encouraging us. Many senior academicians and science communicators are here. Professor C.D. Patil, Professor K.N.B. Murthy, Professor C.K. Manjunath, and many students are also on the Cisco WebEx and YouTube platform. For all of them, thankful from KSTA. Tomorrow we'll be having a talk on Physics Nobel Prize. Nobel Prize awardees work by Professor Bead-Based, Professor of Physics in RRI Bangalore. I request all of you to join at 10.
So thank you very much. So thanks to everybody, and a very, very early good morning to Dr. Ravi, and have a good day, Dr. Ravi. From all of us, ಮತ್ತೊಮ್ಮೆ ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು. ಬಹಳ ಧನ್ಯವಾದ.
Thank you.
ನಮಸ್ತೆ ಸರ್.
Bye.
ನಮಸ್ಕಾರ, ಎಲ್ರಿಗೂ ನಮಸ್ಕಾರ ಸರ್.
ನಮಸ್ಕಾರ.